Thanks, Chris. So before I get started my talk, I just want to give you a link to this notebook that we will get to play around with later in the middle of the talk. So take a picture if you need to. You will be able to run a particular technique I will introduce later on in this Kaggle notebook. So if you go to this link, you can also see the URL for this. It's a publicly runnable notebook. So all you need to do is create a simple account on Kaggle and fork the notebook. And you'll be able to edit, run, and do whatever you like. So you want to take a minute to do this so we can do it later. All right. So does everybody have this information? I'm going to move to the next slide. So you've heard a great deal about a lot of wonderful open data sets, or some of them not quite open, but at least fair data sets. And there's a lot of amazing data for you to take advantage of, and a lot of software to be able to analyze this data. And but one of the downsides of accumulating data across many different sites, and especially from multi-center data sets, is that there is a lot of unwanted nuisance variation. And so today when I talk about site effects, there's usually two kinds. One is nuisance variation due to measurement error. So this is like all the kinds of MR measurement error that Oscar talked about earlier. Creates nuisance variation. There's also other kinds of variation there can be between in large data sets that's based on age, gender, and socioeconomic factors. And today I won't be talking about demographic variation. I think demographic variation has a very complex relationship with your observed data, and the methods I introduce are much more suited for dealing with accounting for technical nuisance variation. Okay. So where does this unwanted nuisance variation come from? So for example, if you look at the ADNI data set, they have a lot of different data from different scanners. So change in make, model, scanner variation is one source of nuisance variation in these large data sets. For example, that was with structural data, you can see the same for like DTI data. This is an example from the core consortium data set that Cameron referred to earlier. And for example, fMRI data as well. So all kinds of MR data, we have a lot of variation in scanner, in the sequence, in the temporal resolution, so forth, and that's one source of variation in your data that's undesirable. There's also, as you saw from QC measures, like for example, with structural DTI and fMRI, motion is a big problem. That's a source of like session-specific variation, but if multiple subjects from the same site have like more motion, for example, it means you also have more nuisance variation in that site. So there's a lot of within subject sources of measurement error that also contribute to site effects. And we can see just from QC alone that different sites have different degrees of this kind of within sessions, measurement error. So a general model for this kind of unwanted nuisance variation. The observed data, you can say, is a measurement of the real signal that we want to get to, and systematic error as well as random error. So unwanted nuisance variation is not just noise. There's a lot of structure in it. It's a very systematic kind of confound, and we can't just hope that we can average it all out. So we're going to examine a few, how to identify this kind of systematic error, and how to at least ameliorate it, if not perfectly eliminate it. And one of the big reasons we want to get rid of this kind of systematic error is that there's a lot of needless heterogeneity between subjects that's created as a consequence of this error. And we're all interested in, a lot of the research questions we're interested in are about understanding individual differences. But we can't understand individual differences, individual biological differences, if we can't get rid of the artifactual individual differences. So that's like a major, major reason that we care about reducing this, as well as just generalizability and just being able to detect your effects of interest. So it's important for a lot of these reasons. So a big overview for today. We'll examine a few different techniques to look at your data and identify, if your data has too much site-specific variation, and also to look at your data to look at whether different pre-processing options actually can help ameliorate and reduce some of these between site variation. We'll also look at different types, classes of methods available today to address this kind of these sorts of unwanted site variation. And then finally, I want to spend some time thinking about, is there like a theory? Are there principles for what constitutes effective and good between site variation? So this is a very, I would say, early and nascent area of research overall. So there's a lot of ongoing development, but hopefully you get a sense of the momentum that's been building up in the last few years with open data sets and now a lot of new methods to kind of address these sorts of challenges. So first, evaluating site effects. Okay. So there's two broad strategies. One class of techniques is all about quantifying the different types of variation. Basically, you want to reduce heterogeneity, and there's a bunch of techniques available to characterize how much unwanted heterogeneity, undesirable heterogeneity is in your data. And that's one way of quantifying site effects. And the other is to actually look at improvements in detecting an effective interest. And so those are, I would say, most... not every paper and every study looks at all of these characteristics, but ideally you want to quantify both of these measures as a way of examining how much site effect there is in your data and whether or not there are things you can do to change and move the needle on these. And I won't have the chance to talk about all of these in detail today, so I'm mainly going to focus on the first. But if you have questions later on and if there's time, we can talk about the others. So there's mainly three different types I want to emphasize. One is a visualization technique that comes from medical statistics. It's the Bland-Altman agreement technique. It's of the top 100 most highly cited papers of the past century. It ranks 29, and it's like widely underutilized. So I wanted to point that out. Another is a clustering-based technique, and the third one might be the one that's like most familiar to you. So getting to the first one. So the idea behind the Bland-Altman technique is that we want to compare, we want a measure of agreement. We want a measure of agreement if you have two different sites and you have identical data or features across two different sites. You want to look at, for example, a healthy population who's getting structural MRI scan in one place versus another, they should look roughly the same. And this kind of measure of agreement is about quantifying the difference, the discrepancy. So broadly speaking, what you do is you plot the difference between the sites on the y-axis and the average between the two sites on the x-axis. And the reason you want to do this is that both sites have some amount of measurement error, so that's why you want to use the average. It's desirable to use the average. If you really want to understand a bit more about the motivations behind this, I would recommend reading their paper, which is the Lancet paper, which is like highly accessible. So when you look for between-site differences here, looking at deviations from zero will tell you if there's some systematic differences between the site. And looking at the scatter, the amount which the points vary, gives you a sense of how much variation, the unwanted variation there is. So for example, here, every point can be a voxel intensity that you're comparing between sites. So that's an example of what you would plot here. So just to see a preview of a paper that we'll study a little bit later as well, here they're looking at DTI-based measurements of a particular set of features between two sites, and you can see that this is the distributional difference of this particular feature between two sites. And when you do the Bland-Altman plot, you can see that this kind of classic deviation from zero is an evidence of there's like being a systematic shift, location shift. And we'll examine a couple of techniques that are able to bring it down. So this is like an example of what's desirable to see and what's how this kind of visualization technique can be used to look for side effects in your data. The second technique is a clustering-based technique. The basic idea is, say you have MRI images on multiple subjects. You want to look at subject-by-subject similarity matrix. So you can use any measure of similarity. It can be based on correlations. It can be based on like rank concordance type measures. You can use any kind of similarity measure used in clustering, for example, to look at how similar are two subjects to each other. And once you create this kind of similarity matrix, the basic idea is, are subjects within the same site more similar to each other than subjects between sites? And if you do this, this is an example on a healthy population. And so you can take advantage of measures used in clustering that basically characterize how much is the between-site similarity different from the within-site similarity. And a simple way of doing that is, for example, look at a Wilcoxon rank sum test statistic between to characterize this, and create a null distribution, for example, using a permutation test. And so you can use this as a statistical test to detect if there is a site effect or not in your data. And hopefully one way of characterizing and comparing this before and after applying some measure of technique is that you can see if you're doing effective site effect correction, what will happen is that subjects are now much more similar across sites. And that is the change that you will see in this kind of technique. So this is also, I think, a very, you're just able to take advantage of good old clustering and clustering methods to detect side effects. And this is something you previously heard about from Oscar too, and there's a number of papers that do this. So for example, you can detect a site effect if you can classify sites based on your data. So there's a really nice, a very large analysis across lots of open data sets by this recent preprint, where they looked at structural measures. I think it was like cortical folding, cortical thickness, and they basically showed that using these measures you can effectively recover most sites. So if you're able to use a procedure to modify your data and correct for site effects, you should see a noticeable difference in your ability to predict sites. So and you don't have to use prediction. You can just do regular old linear regression, ANOVAs, GLM to do the same. So roughly these are three different kinds of measures that look at to quantify your side effects. The other two kinds is to say, let's say you're interested in a covariate of interest, like for example diagnostic group or age or gender and so forth. The idea is if you look for increases or improvements in your ability to classify or detect these effects, that's another measure of evidence that you have eliminated, unwanted nuisance variation in your data. I don't have time to talk about it, so in the interest of time, I will skip this part right now. But if you have more questions, I have additional slides so that we can come back to this if you're interested. So moving on to methods to address site effects, there's basically almost all the techniques out there kind of fall into these three categories. One is sort of intensity normalization type methods. The second kind are location and scale models. They try to center and scale your data in such a way that you're eliminating site effects. And then there's a lot of latent variable models. So the actual methods available for the first kind are, for example, histogram matching and various kinds of quantile normalization. In the interest of time, I won't be covering this class of methods, but I just wanted to put it out there. For the remainder, I will mainly give you examples of methods from both of these kinds. So location scale models, this is like using ANOVA or like multi-level models belong to this category of location scale models. And latent variable models are various kinds of PCA factor analysis combined with regression type methods. Okay. And there's a really wide range of work on a lot of these methods, not just in neuroimaging. In fact, Experimental biology and microarray studies had to deal with these kinds of batch effects 15, 20 years ago. So they're a bit more advanced in having developed methods to address these. But it turns out, a lot of the fundamental mathematical and statistical principles are the same. So there's a lot that we can borrow from these other fields to inform our own and tailor to our own. So a lot of the methods I will discuss are actually come from microarray and RNA sequencing technologies, for example, that we can adopt for ourselves. So I'm going to first discuss a couple of these models in the context of structural and diffusion MRI data and other work that's been done in trying to eliminate especially scanner variation in these kinds of data sets. Okay. So roughly speaking, the kinds of data I'm going to be discussing are where you've got voxel-wise maps of some kind. These could be T1 intensity values. These could be voxel-wise statistics in DTI, like fractional anisotropy or mean diffusivity. A lot of the techniques that I'll present have been applied to these kinds of data. And the main goal is to make sure that these intensities are now comparable across subjects, across sessions, across sites. So a lot of these techniques have been applied in this context. So the first one I'll be discussing are these ANOVA location scale type methods. Roughly speaking, you model each observed intensity at a particular voxel as a function of, P covariates of interest. So this could be something like age, for example, could be a covariate of interest, as well as your matrix Z, which is your nuisance covariate. So for example, an indicator variable indicating that a subject belongs to a particular site would be a known site effect or a site covariate. And you can just solve for this problem per voxel. And your corrected data would basically, you fit this model, you fit the gammas, and you take the residuals. And this is a way of correcting your data. If you don't do this voxel-wise and you do it on the average intensity across the whole map, for example, this would be like a global model. This is sometimes often used, but we'll see some examples later on that show this is not as effective as fitting local models. There was a variation of this technique that was introduced in the context of microarray data over a decade ago. That's been highly, highly effective, and most recently in the last 2 years, it's been applied in neuroimaging as well to great success. And this is called COMBAT. So in COMBAT, it's a slight deviation from the previous model in that it creates both a mean and a scaling variable for each site. And the basic idea is it uses empirical Bayes techniques to fit site-specific mean and site-specific scaling parameters and eliminates that from the data. So the residuals from a COMBAT correction, COMBAT correction of your intensity values would basically look like this. It rescales the residuals and then adds back this component. So you're basically shifting and rescaling your data to make it comparable across images. And so let's see some examples from work done by people who've applied COMBAT to structural as well as DTI data. So here's an example of COMBAT applied to the ADNI data for the cortical thickness measurements from structural MRI. And you can see that there's distinct site scanner effects here, that different scanners have different measures of cortical thickness. And after applying COMBAT, these are now far more comparable. So this is an example of COMBAT in action, and I have, coming back to my little tutorial here, I have a toy data set with QC measures for you to apply COMBAT yourself on this toy data set. So if people want to follow along with me, now's the time to pull up your forked notebook. Is anybody following along right now? Okay, great. Are we ready to, do you have the notebooks open in edit mode? Okay, great. So first, I would just run through the first 4 cells up to cell 4 and just run through them. Basically, you'll be able to load your data, we'll be looking at two QC measures. So this is data from the Pre-Processed Connectomes project with the QAP, which was like an early predecessor to MRI-QC. So we're looking at some quality control metrics on from fMRI data, spatial quality control metrics, and temporal quality control metrics. So we're going to load this data. You should be able to see all the features of both kinds available here. And let me know when able to run them. Okay. Are we ready to move on? Next? Okay. So once you've loaded your data and you are able to confirm, you can look at the features. So I'm going to merge them all into a single table and visualize the site effects using box plots. So you can run the next cell and you should be able to see. So I picked the first cell. I picked ghosting in the X and Y directions. So here is a visualization of this. And you can clearly see what you're seeing on the X-axis is the different sites. And what you're seeing on the Y-axis is the box plot variation of subjects within that site. And you can clearly see, well, there's a lot of differences in the location, in the mean values, and in the variation between sites in both of these measures. So hopefully you've gotten to this part. Yeah.
We're getting a directory in the folder. Is this still there?
Yeah. I picked Kaggle because I would hope that would minimize any kind of startup costs because the data set is there and the notebook's there, so it should run smoothly. Yeah. All right. Okay. And okay, now we're ready to run Combat. If you're interested, you can always do help Combat if you want to see the functions that are built into this notebook. This also comes from the Bioconductor SVA package. So this is an implementation. This is the gold standard implementation for Combat. So if you're ever interested in running it in R, for example, you can go here. And you'll find Python variations as well. The next cell, what we're going to do is, so Combat takes in data where the subjects are the columns and the rows are the features. So you actually have to transpose your data a little bit and before running it. So you can run Combat. And then we can now visualize the changes after running Combat. So the same ghosting that we saw before, now you can see that, definitely the means are very close to aligned. You can barely tell. They're all overlapping confidence intervals here. And just to see an example of before and after, this is what the results look like before and after. And one of the things you have not visualized here, but I would encourage you to check out, is to actually go back and look at the fitted parameters, the gammas and deltas that Combat fits, because this actually tells you the transformation necessary for each site to transform it to look normal. So conceivably, if you've already got, for example, if your university, for example, has contributed data to one of these public data sets, you can probably get an estimate of what your data looks like in terms of these gamma and in terms of the Combat parameters, and potentially use that to compare it for future new data sets. So that's the kind of thing you might be able to do with Combat. Yeah. So you can create like pre-built transformations for every site. So if you're a site that's contributed a lot of data, you might be able to do this yourself. So this kind of location scale-based techniques work very well when you've got some categorical covariate like the scanner, make, model, and so forth. So it has to be known covariates. Known covariates meaning, this is like a known fixed quantity. There's no uncertainty that some subject was scanned by a particular scanner. So for things like that, Combat is great. But one thing to keep in mind is that it treats each feature separately. So here you're applying a separate model for every voxel. You're applying a separate model for every feature. And so it's not taking into account the joint distribution across all features. And one of the deficiencies of that, that's why I say, this kind of technique is great for first-order statistics. But for second-order statistics, so for example, if you're looking at measures of brain networks, either functional data, morphological networks, these are all second-order statistics. And if you want to preserve the joint distribution across all second-order statistics, it might be undesirable to apply Combat on, say, every correlation value, because the resulting matrix now is no longer a proper correlation matrix. So be careful on what sorts of things you apply Combat to. But one general rule of thumb I use is that you want to use it on as close to the raw data as possible, because that's where it's most effective. So we're running a little bit short on time, but I want to quickly mention a different class of methods. These are latent variable models. And here in latent variable models, you might have, this is your data again, you might have some covariates of interest like age or group differences or something like this. And the idea with these class of models is that there's some unknown set of latent covariates that explain all the unwanted variation in your data. So this could be a combination of various kinds of measurement noise. It could be the motion, it could be the thermal noise, it could be scanner-specific variation. So basically here, you're not using any explicit covariate. This is like latent covariates that explain all the unwanted variation. And there's a lot of methods that follow this general model, but roughly speaking, there's going to be some technique to infer these unknown W's, the unknown latent covariates. And once you infer, you have an estimate of these W's, you now use regression to regress out these from your data. So that's roughly how a lot of this class of techniques work. There's many variations of this. And a lot of them hinge on the idea of what is the relationship between W and X. If W is independent of X, that's great. But if the W's and X's are confounded with each other, then different methods have different, the mileage will vary based on different methods, and you might need to use more sophisticated notions. So I'm going to look at some studies that actually compared a bunch of different methods, including latent variable models, so you get a sense of how well they perform. So this is a recent paper by Fortin et al. In Neuroimage. It looked at DTI data, so it was trying to harmonize scanner variation and DTI. So it's applying correction on 3D maps of fractional anisotropy. And we are looking at the Bland-Altman plots that I mentioned earlier. So what you see here is the raw data of how the data varies between between sites. So certain kinds of global scaling techniques, they center the mean, but there's still a lot of variation. And this is a class of intensity normalization techniques that also, they roughly center the mean, but there's still a lot of variation. And then there was a whole bunch of latent variable models. The Ravel and SVA are both latent variable model techniques, and Combat. And this technique uses CSF voxels as control voxels to model your W's, to model the latent confounds and eliminate from the data. This doesn't make any assumptions. SVA doesn't make any assumptions at all. And Combat we just went through. And as you can see, Combat does exceedingly well in this kind of data. Like, you've basically eliminated a lot of the between site variation. It's almost, it's substantially reduced. So it's like highly, highly useful. And SVA is a close competitor here. So what's really nice about this paper is that it looked at a range of different measures of both quantifying the side effects and quantifying the improvements from side effects. So they looked at 2 sample tests to say, on each voxel, to say, am I able to distinguish between sites? on each voxel, and they look at the percentage of significant voxels. And what you find is all the three, the final three measures, Ravel, SVA, and Combat, barely have any voxels that have detectable site differences, which is understandable from looking at these blend-Altman plots. But what they also looked at is the ability to, they quantified the effect size of being able to detect age-specific variations in the voxels as well. And what they found is that, Combat was able to do the best on this. So it increased the effect size of interest, or as an example, it increased an effect size of interest. So that's another highly desirable way. Like, based on this, you couldn't tell which of these was performing any better. They all look similar, right? All the three. But by looking at the effect size of interest, you can see that some are doing a better job than others. And one of the reasons in this particular data set that Combat did much better than SVA is that SVA makes some assumptions about the relationship between your latent variables and the covariates of interest. And in this case, age is the covariate of interest, and the site and the age are confounded with each other because of this. There was a slight SVA kind of like lost some signal here compared to Combat. Okay, so the experimental biology genomics literature has done some very extensive comparisons of a lot of latent variable modeling techniques. And in particular, they've looked at to what extent are your latent covariates and your covariates of interest, independent, moderately confounded with each other or extremely confounded with each other. And different methods behave differently under these circumstances. So you really need to understand the structure of your data, the structure of the nuisance variation, before you can figure out, which methods are going to work on your data. So as an example, I think some of these techniques have not yet been applied in neuroimaging, but you can see that some techniques do much better in dealing with confounded latent covariates than others. And so you have to be kind of cautious in looking at latent variable modeling techniques. Okay. So that was structural data. We'll now look at an example from functional fMRI data, and particularly in resting state. Okay. So roughly speaking, let's say you have data that consists of time series or volumes and brain regions or voxels. Okay. And one message I want to say is there objectively you can find that there's some kinds of nuisance variation artifact removal techniques for fMRI that do a much better job of eliminating site-specific variation. So it's really useful to actually apply a few different pipelines. You can do this with publicly available data sets like the Abide1 preprocessed connectome data because you have different sets of pipelines. And I don't think anybody has actually done between site comparisons, like methodology comparisons on who's better able to eliminate site effects or not on this data. So that's like something that's definitely, you can do it if you want, especially using data from this top. Okay, so we'll just look at a simple example of how you can look at whether a particular artifact removal technique is doing a better job in eliminating site effects. Okay, and so basically the model for this is a kind of latent variable model for fMRI. So basically this is your observed fMRI measurements, and you can say U is the true bold signal that you're interested in, whereas W represents all the unknown latent physiological motion confounds. Okay, so this is a thing you already do. You already do nuisance regression, but a different way of looking at this problem is to transform them into the correlation matrices or the covariance matrices from these variables. So you can think of the observed correlation matrix, which is the correlation matrix you would get if you were to just look at the uncorrected fMRI measurements. And you can decompose it as a function of the true bold correlation matrix and a nuisance correlation matrix. So an example of what this look like is, here's the observed sample correlation matrix. Let's say I use as a nuisance, I use like motion or anatomical composite correction or any of the nuisance variables as W here. I can now compute each of these quantities I can estimate them on the data. And I can now decompose the denoised correlation as the observed correlation minus the nuisance correlation. So you can see how the structure of the data comes out. So this is just a different way of visualizing what a lot of your pipelines already do. Okay, and you can look at how basically the changes in the intensities of these values are often related to are a source of side effects. So we're going to come back to this visualization that I mentioned before, which was comparing the strength of the correlations, subject by subject similarities within and between sites. And this is what it looks like for the observed correlation matrix, which is uncorrected. You can see this has substantial site effects, okay, using our statistic. You can also see that a lot of the structure of how the subjects are similar or different from each other is actually captured by their nuisance correlation matrix. And if you look at the uncorrected version, you've now reduced between subject heterogeneity. Okay, so there's an example. See that actually your nuisance correction reduces site effects. Okay, it reduces unwanted heterogeneity. So you can apply this concept to compare different pipelines or methods to see how well you're doing, and choose to go with the pipelines, especially if you're combining data across multiple sites. Choose to optimize pipelines depending on the analysis of interest that achieves this kind of outcome. Okay. So So there's a lot of other evidence from the literature that good nuisance correction is important for dealing with site effects. One class of studies have looked at between site reliability. So the same people are scanned in different sites across different scanners, and this is a great way to do methods development for deciding which methods are superior versus not. And so there's been some really nice. I don't have time to go over them, but there's been recent work analyzing this kind of information and looking at basically reducing session effects, reducing when you average across sessions, you get better between site reliability, which is telling you that site-specific nuisance variation is a problem and is a contributing problem. There's also been other evidence from the literature that shows how, for example, better motion correction improves effect size of interest. So there's a lot of evidence that session-specific contribution to side effects is a big problem in fMRI, and the better noise correction you do, the better you eliminate side effects. Okay. It's not exhaustive, but it takes you a long way. And definitely this is a very open area of research. We've barely scratched the surface in terms of adapting a lot of the methods out there to correct for side effects in fMRI. So I want to take the time now to take a step back and think about, well, is there any theory? Are there any guidelines for deciding what constitutes, a good nuisance covariate? What constitutes, when do we know we're achieving good site effect correction? Are there any overarching guidelines? And I think we can borrow a lot from epidemiology in learning to deal with this. So roughly speaking, you can create a causal diagram, a very toy causal diagram. Says, let's say X is the true biological signal of interest that you never observe. What you actually observe is Y, which are your measurements that you make from your MRI data. And sometimes you have, a known covariate, like, for example, that a subject belongs to a particular, was scanned by a particular scanner. That's a thing that's known, that's Z. But then there's a lot of unknown, unobserved variation or confounds that are affecting your measurements. So this is like a toy causal diagram of what's going on. And our goal is to maximize we want to detect this X to Y. We want to maximize this. And we want to eliminate this. Right? That's our goal in doing all of this. So just examples of here. And there could be other complicating factors. There could be that there's other hidden confounders that both contribute to the biological signal of interest and contribute to the measurement error. So age, for example, would be a candidate for a hidden confounder. Similarly, in fMRI, we could have a similar kind of paradigm. So basically, this is just a toy model of what's going on in a lot of our data. right? And so epidemiology and experimental biology, they formulated this concept of what's called a negative control. So an ideal negative control is something that only captures, is a good proxy for, basically, the measurement error, but is completely uncaused by the biological signal of interest. Okay, so this is an ideal, so if you are able to, the more ideal your negative control is, the better you can just use, for example, regression and add your negative control as a variable you want to control for, and you're able to eliminate side effects. So a lot of the work that needs to be done, or the theory to guide us, is to figure out what does the structure of your problem look like? What is your X? What is your Y? You know, are you in a situation where you've got a perfect negative control? Right? And so, for example, examples, we already do this in some sense, but there's also controversies. And so I think this theory of negative controls from epidemiology can be used to actually analyze all the various types of nuisance covariates that we widely use in our in neuroimaging practices to really decide what's the structure of our problem, what are the hidden confounders, you know, do we have a good negative control? So, for example, you might use white matter and CSF. Those are one of the top, I think, most popular negative controls used in the literature. This is actually used by a number of techniques that try to normalize intensities in structural data in DTI. We also use, white matter signals or externally, cardiac respiratory signals, for example, as negative controls in fMRI. So there can be two kinds of questions. One is, are you inadvertently capturing some X in your negative control, in which case that's not a good negative control. The other could be, are you actually capturing, does your negative control capture all of W, or is there residual unexplained stuff? So it could be in both directions, right? I think this diagram also sort of helps capture why it might be desirable from the perspective of measurement error to correct as much as possible at the source of measurement, at the source of observations. You don't want to try to correcting for something like age or movement after you've created a lot of downstream variables might be undesirable because you have now lost the opportunity to maximize this effect size between X to Y. So I think this is something, this is an area we can borrow from to actually really understand, how well we're doing and where to advance and improve our methods. So just a brief summary. Like ultimately we want to look at, what sorts of models for every biological phenomenon of interest and modality of interest. We have to think carefully about what does the measurement error confounding model look like, and how well are we doing in modeling or achieving perfect negative controls. And the other thing I want to highlight is that we're dealing with a lot of with data where the number of nuisance variation, actually your W can actually be lots of variables. This could easily, maybe it goes into the tens in fMRI. It might even go to the hundreds. So there's a lot of empirical statistical issues, which is if you've got too many variables here, then you've got too many parameters to estimate, and you might not be able to do a good job estimating them. So there's a lot of statistical issues too in doing these sorts of things. So those are some limitations and caveats, and I highly recommend actually a lot of papers from the cleaning fMRI issue in Neuroimaging that came out last year, which has, I think, a lot of terrific papers giving you guidelines on thinking about these sorts of issues. And I guess, sorry, I was supposed to have that. I don't think I have that. The data reuse question. Submit your questions here. So anyway, I think there's a lot of exciting developments. I think we're still at the cusp of actually learning how to eliminate batch effects and side effects from our data. I think there's a lot more that can be done. And I'm hopefully, based on recent work I've been doing, I'm hoping to curate a resource on both literature and resources on correcting for side effects. So this is not yet populated, but it will be, so keep an eye out for that. And that's it. Any questions? Yeah.
thank you very much. Do we have any questions for this talk in particular?
It's very stimulating talk. I think, but it's a little bit issue is about how these corrections will affect the final results, such as using the abide data set to look at the difference between autism and normal case. So how these corrections will improve or...
Right. So I think, so I will say that there has been, there was one recent study, I think this paper by Abraham and folks, they didn't compare procedures for eliminating side effects, but they definitely looked at a whole range of measures where some were able to achieve better diagnostic prediction than others. So it's definitely, I think, like I mentioned before, it hasn't been done, but it's definitely a sort of thing you can do, which is compare methods that are eliminating unwanted heterogeneity between subjects, and then also simultaneously look at, are you able to achieve better diagnostic accuracy? And for sure, I think there's bound to be some insights that come out of an analysis like that. I just, as far as I know, I haven't seen that particular analysis be done. Yeah.
So related to that, that hasn't been specifically done with abide, but Cha Ganyan has 2 papers where they specifically paired several different ways to standardize against side effects and their ability to serve sex prediction or distinguishing sex and age in there.
Great. Thanks for bringing that up. Yeah.
So this concludes the talks. Let's thank Marjan once more. And now we're going to have a more general Q&A.